Oh, come on. Okay, good. Let's start. So we're going to start with the secretion systems. I guess so. Okay. Oh, come on. Show up. Okay. Secretion system one. So I'm hoping I will have enough time. If not, we will finish next slide. But so if you remember from Tuesday, we talked about protein translocation. So now we are still in the same unit. We talked about moving something across the membrane, just as you expect, more complicated. Well, maybe more complicated. That's how you look. But there are different, and there is a difference between translocation and secretion in terms of physiological mechanisms. It will be clear in a few minutes. But we will cover each bit secretion systems. Which is very specific by some landmarks. So I will tell you what are those landmarks and what you have to know for your exam. There are a few mechanisms that I will clearly ask, and I will, of course, tell you why it's important to know that. So again, this is just an overview, and this would be type 1 secretion system, which I'm going to cover first, as you expect. So what would be the general structure of type 1 secretion system? So as you can imagine, one thing that will be associated with the majority of these secretion systems would be some high energy bonds. So you have to invest some energy to transport something across the membrane. So the very first component of type 1 secretion system would be so-called ABC transporter in inner membrane. So this part will be most responsible for energizing the transport. Next one in line will be, as you can imagine, something that will connect inner to outer membrane, which will be membrane fusion protein or MFP, simple as it is. And that MFP will connect your ABC transporter in inner membrane to outer membrane transporter, which will be TOLC. So as you can probably guess now, we're talking about gram-negative system. Gram-positive is completely different. So that means that type 1 secretion system is present in gram-negative bacteria because of two methods. So specific substrates will be transported through this type 1 secretion system, and it's going to be similar for the rest of them. And of course, there must be specific targeting mechanisms which will happen into the inside of cytoplasm that will target those proteins, so proteins that are targeted that are meant to be exported to type 1 secretion system have to be modified so it can be recognized by all the machines. This is SEC-TEC independent secretion systems, and it will become more clear when we compare this with type 2 and 5 secretion systems, which are SEC2 and 5 dependent, which means you have to have protein exported first through SEC system before it gets into the whole secretion system machinery. But this one, we call it independent. Only two of them are dependent. And the first example for type 1 secretion system that we will cover is this hemolyzing transport. So what this system does is, as the name says, it transports this toxin hemolyzing through the inner membrane. So it has BND, HYL, Hill BND system, and it has TOLC. TOLC is very common part of type 1 secretion system. If I ever ask you what is the landmark for type 1 secretion system, you will tell me outer membrane protein TOLC. So that TOLC will be standard, and then you will have, in many examples, you will have variations, like there will be usually inner membrane component, which in this case is it's called HLYBND. And then you will have membrane fusion proteins. So in this case, B would be inner membrane, which has ATPase activity. This would be analog to ABC transporter. So when I mentioned on the previous slide, I told you about the general structure of type 1 secretion system, like ABC transporter, like fusion protein and outer membrane protein. So in this case, this ATP activity transporter is ABC transporter HLYB, and then you will have membrane fusion protein, which will literally be like a connective linker in between B and tall C, which is in outer membrane. And the transport is, as you can imagine, ATP dependent. So, what is the mechanism of this transport? So, as I said, proteins have to be somehow modified to be targeted um, for the one secretion system. And don't get confused, this is not the same type of N-terminus signal sequence that we talked about last class. So, but the process is very specific. So basically, the substance that is meant to be exported through type 1 secretion system um, is recognized by membrane fusion protein. So that membrane fusion protein that connects the inner membrane and outer membrane is what actually, so its N-terminus is recognizing substrate for transport. Now, when substrate is recognized, once when that N-terminus from connecting in case of HLY, the component, once when substrate is recognized, it's going to be brought up to the ABC transporter, which is the HLYB in the case of hemolyzing transport. So again, membrane fusion protein recognizes the substrate, and then once recognized, then it's going to be bound to the ATP transport, ABC transporter that has ATP hydrolytic activity. Once when substrate is bound, again, recognized and brought up to ABC transporter, that will cause the conformational change into membrane fusion protein. And membrane fusion protein that now changed conformation will interact with TOL-C, which is outer membrane protein. So basically, this binding of the substrate by ABC transporter and the membrane fusion protein is a recruitment signal for the outer membrane protein, which is toxic. So all that process from membrane fusion protein recognizing substrate and binding to ABC transporter, that binding is recruitment signal for often toxic. It doesn't have to be toxic, but it's very often called toxic. And then TOL-C, which is again embedded in outer membrane, will create the pore for substrate passage. Now, when that pore is created already, so it's ready for substrate to be exported, ABC transporter, again, in case of hemolysis, that will be HLYB, that transporter will perform hydrolytic, ATP hydrolytic activity because that energy will be used to push substrate through TOL-C pore. So again, mechanism might be a little complicated. I usually think that the next example of RTDX toxin is more easy. This one might be a little more complicated, but this is a nice summary, at least I think it's a nice summary of the order that we went, how everything is happening for hemolyzing export. So recognition of hemolysis by membrane fusion protein, binding to the ABC transporter, which has hydrolytic activity, conformational changes in membrane fusion proteins, activation of outer membrane protein and pore forming, and the last but not least, ATP hydrolysis. Again, this might be a little complicated. The other one will be, in my opinion, much easier. So before the protein is brought up to the structure, before all complex is formed and substrate is ready for export, B and D components, like membrane fusion proteins and inner membrane proteins are open, like so they are separated. But once when protein arrives, it will bind, dock to the ATP component, and once the protein is settled, B and D come together, which is followed by TOL-C association. So this is from paper. It looks more like a story, but it's literally that order of events I just told you. Can you split them up? In my opinion, the other one, marked toxin, is much easier. It's from Vibrio cholera. So when I was a TA, I introduced this part because I thought it's going to be hard for students to understand hemolysis. Turns out it's not. Hopefully it's hard, but it's much easier because the paper had very nice picture which shows how there are inner membrane components G and E, and then there is another one D, which is like a membrane fusion protein. but it's actually also part of inner membrane, and it extends right to the 12C, which is in outer membrane, and you can see how all toxin is going to be exported. So, and this is the another process that I really like, and I found it very easy. So the marked toxin is toxin from Vibrio cholerae that is exported through type 1 secretion system. So first what marked toxin has to do is to interact with inner membrane component, which is so this RTXC is in inner membrane, but it really kind of plays the role of membrane fusion protein because it extends to the source C, if you can see how it looks in the picture. Uh, Next one, when this D component, which again plays the role of membrane fusion protein, interacts with the substrate and interacts with the substrate with its N terminus. So it finds the substrate toxic. In this case, substrate will be exported with its N terminus. So again, D plays the role of membrane fusion protein here. Now, when it found it, like they found the toxin, that toxin will now closely interact with the rest of the inner membrane components, which are B and E. So RTX, B, E, E are together in inner membrane, but B kind of stands out a little bit, and B actually finds the substrate inside the cytoplasm. So now when toxin interacts closely with B and E, that's number 3 and 4 on the picture. All that interaction between B, and E are actually going to be triggered for this tall C pore forming, which will be the passage for your substance. Of course, as you can imagine, this needs to be energized, and both B and E components are going to perform hydrolytic activities because both of them are actually ATPases. So if you remember hemolyzing, it was only B. Here you have B and E like a two pass, like a dimer that will complete hydrolytic activities, in this case, 2 ATP molecules, and that hydrolysis will actually push martoxin through the C. Once when martoxin is outside, it reacts with calcium, it reacts with calcium ions, and those ions will help in cooling the toxin that will later on be delivered to the cell. So this is all that structure. Which proteins will be exported through type 1 system? So, I mean, you can already guess that most of these proteins are toxins. They're pathogenesis related. They will be enzymes that will attack host cells, so host cell proteins like proteases, nucleases. There are various types of non-pathogenic, but what I always found is that it is somehow related to toxin and that's why we know it's from pathogenic organisms. So this will be all for type 1 secretion system. Next, yes, Jeff. But because I had to share another one. Yeah, no, I'm going to. I know what I'm doing. Nobody's. I know. I learned. If I didn't, Marisa is there. She will tell me. Let's see. Well, comparing to me, everyone is tech genius. So there is no question about that. I'm just not. Let's see how this thing works. Come on, come to mom. Slideshow. From beginning. You see, I got it. Okay. So with this, we're going to move. So these two mechanisms of type 1 secretion system, yes. Yes. Please. That's before the protein binds to the ABC. Open up the liquid. So actually, and not connect when they remove after the bottle. So think about that as a mechanism, like they're not fully separated, but they're opened up because they have structure open to accept substrate. That's why we say open up. So once when substrate gets recognized, they literally change conformation, they come all together, So it's not really closed structure in terms of like a closed not doing activity. It is they're brought up together. They change conformation. That interaction actually leads to formation of both. So we say they're close. They're just closer together. They're not like when they're not binding to substance, they're separate. But then when they bind, they change conformation and that ends up. So just you have to think 3D, like how you process. And you can talk more about that, not to your head. In my head, that's all was. No, but it is not clear to you that. Okay? And are you going to okay. I mean, it's not going to become easier, so better ask. Because the next one that comes is going to be sex dependent. It's called GSP. So you're going to have a lot of mechanisms. It's called GSPDE, and there is this one student found the most complicated. I mean, the process will still involve ATP, but there will be that periplasmic event, and that's why it's sex dependent. So if you thought type 1 is complicated, wait to hear type 2 secretion system. So it is cell-dependent, which means that proteins that are exported through this system have N-terminus that will first be exported. Very often components of these systems are exported through second, third, or a translocation system. But what this literally means is that proteins that are exported through this system have to reside sometime in periplasm. So they're not, it's not continuous structure like in case of type 1, 3, 6, and 4, 5, 4 actually, and 7 and 9. This, 2 and 5 have that periplasmic event where protein has to reside in periplasm a certain amount of time. And the reason why is because in periplasm that N-terminal signal sequence is going to be removed, and then after removal of that signal sequence, the structures that are usually similar to pillins are going to extend and push protein through the cell, well, through the periplasm. So it's like a piston-like, we say piston-like systems that drives ATP hydrolysis. So it's slightly discontinuous comparing to type 1 sequence. So proteins will be fully folded and active after removal and terminal signal sequence in periplasm. So that's what distinguishes this type of secretion. So it has to be persistent. You have to remove that N-terminus. Very often you will have chaperones involved, and those chaperones are usually active in periplasm because they help folding the protein after removal signal sequence, unless you have a TAT-dependent system, because TAT-dependent proteins are fully folded in cytoplasm. Here you also have outer membrane structure, so it's still similar, like inner membrane ATP hydrolysis, something that connects an outer membrane pore, but it's not TOLC. This one is quite different. So TOLC is specifically landmark for type 1 secretion systems, and through that port, your proteins will be exported to the outer membrane. So as the protein for export comes to the pore, there will be that polymerization of proteins that are similar to pills, and then that polymerization will actually be like a piston that pushes proteins outside of the cell. And the example is this GSP, GSP signal, the GSP signal process. So there are various GSP proteins. There are two parts that I wanted to talk about here. And that's why it's slightly more complicated. Well, not too much, but you will have to know that it is GSP based. So the main component of outer membrane protein is GSPD. So again, not TOLC. We have completely different structures, so it's quite diverse. So GSPD is the outer membrane structure. So GSPD is going to be is going to be exported to the first periplasm and then outer membrane through SEC translocation system. So it needs to go from cytoplasm through inner membrane and then through periplasm to be embedded in outer membrane. Um, the next one that will also be exported through SEC channel will be another outer membrane component, which is GSPF. So you have now S and D that are like analogs to the TOL-C4, and both of them are exported through SEC channel. So S and D are attached to each other and embedding itself into the outer membrane, forming a pore, because that's a platform for substrate export. So once when S and D are established in outer membrane, um, the inner membrane components, because now you have building literally all this system through SEC export. So now first you build like outer membrane, and now when that's established, it's like a docking, this time in outer membrane. Now inner membrane proteins can be exported through SEC channel. And this is where part two comes into the game. So another GSP proteins, which are of course not DNS, but they're called LMC and F. How easy will be to memorize? But if you miss one letter, I'm not really going to be picky about that. But it's important for me what I would like you to know is that in this case you have or that is not TOL-C, it's GSPD that will be important. It's like 2S. And there are different types of GSP proteins, which if you miss the letter, I'm not really going to be. The pool is important because it's quite different than TOL-C and it's going to be assembled first through SEC. So first will be exported through SEC channel and then will be assembled in outer membrane. Then inner membrane component again assembled through SEC channel and they cross periplasm to contact the outer membrane. So they go through SEC channel, they contact outer membrane, that they assemble into the inner membrane. Last but not least is that you can imagine involved in ATP hydrolysis, simple because you need to have still that component that will provide energy. So GSPE, and this is important. So it's important to know that this one I do want you to know. GSPE is ATP hydrolysis, it's ATPase, so it performs hydrolytic activity, and it will bind to the inner membrane protein to complete the structure. GSPE binds from the inside of the cell. There are other GSP proteins that I'm not really pointing out here because it's too hard to understand and it's too hard to memorize. General structure is similar. So if you think about what general structure of type 1 signal systems, so what do you have? You have outer membrane pore protein, you have membrane fusion protein, and you have ABC transporter in inner membrane that has ATP hydrolytic activity. So what do you have for type 2? It's not much different, just I mean, it is different in the morphologically, but functionally, you still have outer membrane platform. Just in this case, it's not called C. We just saw that it's called GSP and S. You still have ATPase, just it's assembled from inside. It's not the integral membrane part. It's just floating and comes up after assembling both components. Of course, you have inner membrane, pretty much similar as you had for type 1 signal system, just it's a different type of focus, and the assembly is done after translocation through SEC system, and you get this pseudoparticles. So this pseudoparticles is a characteristic of the signal execution system because that polymerization and depolymerization is like a piston that drives up the protein. And you probably remember something similar that we talked about. We talked about pills, type 4 pills when we talked about thermophilic structures. So this is very similar. Now, what's the mechanism of this transport? Again, different but similar. So stay with me. It's not easy, but you will get it. So you synthesize now protein for the export, so that has to have an terminal sequence. And based on that sequence, proteins are targeted through inner membrane. Again, this N-terminal signal sequence is the one that will target protein to be exported through second TAT system. So N-membrane components and proteins for export are both exported through SEC system. So when exported through SEC or TAT signals, SEC or TAT translocation systems, there will be peptidase in periplasm that will cut out that N-terminal signal sequence in periplasmic space. And in periplasm, protein will bind to GSPD. We talked about GSPD, that will be outer membrane pore. And that binding in periplasm to the outer membrane will stimulate ATPase activity of GSPD. That will be so that ATPase activity of GSPE will be followed by additional proteins that will form that piston, pseudopills, and that pseudopills will push the structure through channel in outer membrane. Similar like ATP, like type 1 secretion system, it will be, as you can imagine, in gram-negative bacteria, because anytime when you have this inner and outer membrane, you guess that it's about all gram-negative bacteria. Toxins, sometimes cytochromes, but what I usually saw is that it is related to pathogenic organisms, and most of these proteins are studied in pathogenic organisms like hydrolytic enzymes that pathogens export to attack host cell. The type 2 and type 3 secretion system is SECTAT independent. So there is no that periplasmic step and TAT involved. And this one is from what I know, when I studied, it was very much famous in pathogens, unlike type 6, which is also present in social bacteria. But type 3 is very common in pathogens, and it has that syringe-like structure that really to me, this one and type 6 really much looks like a phage structure, and you will see now why. So landmark for type 3 secretion system is this syringe-like structure. So that connection between, so in order to attack host cell, pathogen has to attach to the host cell. So these pathogens have to do that adhesion onto the host cell, and first that adhesion has to happen, and then you will get injection of hydrolytic enzymes or proteins or toxins that pathogen will inject through this system into the host cell. So what usually people say that this is similar to flagellum. I am always confused between this one and type 6 secretion system, because one reminds me on phages, and this one also has phage. To me, it looks very similar to phages, but type 6 is way more resembling phage structure. This one, papers will tell you that it resembles flagellum. It's very similar to flagellum. But proteins that will be pushed through type 3 secretion systems are mostly related to pathogenic structures. So it is like similar to flagellum. So you will see structural proteins that will build base in a rod and needle structure. So that needle structure is or syringe-like structure. So that's what is a landmark for this system. So why it's similar to flagellum? Because it has the base, it has the inner membrane rod. So pretty much if you remember flagellum and all those rings, that's what it reminds, but it's slightly different morphologically. Another component, so these are structural, are effector proteins, so that will be nothing but hydrolytic enzymes, like molecules that will be injected to host cell to promote the disease. It's called, all those proteins are called injectisol. And chaperones, which are proteins that help in folding. So chaperones are protective proteins that prevent aggregation, prevent usually degradation of injectisol proteins, because they have to be in fully folded state in order to promote disease. general structure of three complex. So as you can see, it really reminds on flagellum, but the needle is something that is not like in flagellum. So that needle is actually very big landmark for this type of institution system. And again, obviously, this is gram-negative bacteria. The whole structure of that distribution system first will be the needle, and you have to have regulatory and effector proteins and chaperones. So that's what we talked about with those three types of protein. And transportation machinery, which is literally something that goes through the outer, through connector and through outer membrane. Very so this actually looks like this is the picture that shows how similar it is to flagellum. So my apology, this is really not like phage. Type 6 more like phage. Uh, so this would be flagellum. So if you think about it, if you see this inner base, like the inner membrane structure base, that's pretty much the same. But then in flagellum, you will have that hook and early and late, and you will have flexible structure. Middle is also flexible, but it has a different evolutive role. So middle is here to set the whole cell, and you inject proteins that will promote disease, and flagellum in bacteria is there to read motility. So uh, again, just characteristic, the ring-like structure that spans inner and outer membrane, very similar to flagellum, hollow structure that hollow structure which material is pushed, very similar to flagellum. But one of the differences, except that it's used for different principles, is that flagellum is using proton multiforce for movement, and this one is using ATP hydrolysis, which means it has some ATP machinery associated. The example are proteins that are injected from Yersinia. It's a black plague. So this is very simple mechanism to comparing to what we as you can see, there is ATP component in the inner membrane. So one of the first steps is to have pathogenic organisms coming into the close association with the host cell, because adhesins have to be synthesized. So adhesins are outer membrane proteins from Yersinia, and they are secreted to help attachment to host cell. So once when adhesins are secreted and when two cells, like host cell and pathogen cell, come together, that is a signal for yolk proteins, which are part of injectosome. So once when yolk proteins now have to be injected into the host cell. So what you need to get here, you have to get ATP hydrolysis that will push these injectosome proteins through the all complex. And the length of this needle, and again, needle is the landmark. So the length of this needle is genetically determined, and it's based on the length is based on the distance between host cell and pathogenic cell. So that is something that is that's genetically determined, because it will depend on, so the length of the needle will depend on how many or how much adhesive proteins you have to secrete to get closer attachment. Another example, so yolk proteins are from Yersinia. Pseudomonas aeruginosa, you probably heard about this, and this one is, so historically I like to ask this question because to me it's very easy to make analogy, but students found it hard. So because you are having an analogy and how cool it is. So it contains proteins X. It's called X. So I really like it. XADEC. That's how proteins are called. And there is a very specific mechanism how Pseudomonas performs this export. So when there are no infection, so there's not going to be adhesins in bacteria, Pseudomonas secreted, and then this XE molecule is going to accumulate in bacterial cell. Now, Xe molecule, and I will make analogies because it's going to be easy to memorize. So X is, think about Xe protein when there is no infection being trapped in a bad relationship with Xc. So I guess we also have that. So Xc is trapped by Xc is trapped by Xc, so very bad relationship, but you can actually, again, very easy to memorize. So because E and C are stuck together, again, C holds E, there is no adhesins, there is no injectism secretion, nothing can happen. No career progress for Xe. So why is that happening? Why is Xe ending up in a bad relationships? Because of XA problem. And XA is the key point here. So XA is also stuck in a bad relationship with XD. So think about A and D as a vitamins that are present in milk. That's how I memorized. So C and E are in one relationship, A and D are together in another relationship. So, but XD actually really likes XC. So they have affinity for each other. But because C is in bad relationship with complex C, nothing can happen. So A and D are stuck together, C and E are stuck together. But D also can bind C. Why it can't? Because C is trapped by E. But when adhesins get synthesized, so initiate the fact, so when they bind whole cell and adhesins are synthesized, that's the signal for D to get released from A. So it's like a breakup. So adhesins are like, wow, I'm stuck in this relationship. I have to leave. So now when D is released from A, and that happened when adhesins got synthesized. Now XD goes to XC and says, hey, I have affinity for you. Let's get back together. And XC is now trapped by XC. So it's literally like a changing of interactions. So XA gets released by XC, so everything is great. What that means is that now free XA can go and activate transcription of genes for injectosomal proteins. So it's all about changing the patterns so everyone is happy. DNC can be together because they like each other, they have affinity. A is good to go and do its own transcription, so everyone is happy, everyone is busy except E. E was a bad guy. Does that make sense? That's how I memorized. Will never forget. And this unresolved issues with type 3 secretion system, take this with a grain of salt, simple because I did not update lecture, and I'm pretty sure that these questions already got resolved. I'm not really sure. I always forget. Right? So, evolution, yes, Rada.
Just on the last slide, the second part, it says XA is released by XC.
Uh-huh.
Because XC was connected to XC.
Mm-hmm.
Would XC
Let me see. I'm getting. It's not. Maybe I didn't say that right. So the XC. So A and D get together. C and E get together, right? So once when you have, so what you need to have XA uh, free to activate transcription. Which means what I am trying to say here is that XC releases XA, so it doesn't bind it. It's like, so in the first complex, A will be with D, right? I know what um, I think E should be corrected to A. I apologize for that. I think that, thank you for, please write me that down. Okay? No, okay, sorry. What did I miss here?
I think it's X, A, and the second bottom part, that's supposed to be X, E,
Yes.
X, right, X, T,
Yes.
and then X, A, because
Yeah, yeah. So A and D are together, C and E are together. I think I just remind me to correct this on the slide. The point is that A and D are together, C and E are together. When adhedins are synthesized, the whole point is to get, so D and C have affinity for each other as well. But D is with A. When adhedins are synthesized, D can bind to C, and then E will release C, but then A can be released by D to go and activate transcription. And I will post additional explanation so you guys don't get confused. Thank you for pointing that out. I have to correct that. Did you? Yes, please. And I will announce that so you, because this question usually comes to the exam, so I want you to all be clear on the left. Yes. Say it again. Yes. 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 I I call it X. So I know it's EXS, but I call it just X. It's easier for me to pronounce. But you can like X will be fine. That's how I memorize. You can imagine it. So does that make sense? So A with D, A with D, C with E. What we need to get A to be released from this, so to be able to activate transcription. So the key factor here is A. Does that make sense? Okay. And again, unresolved issues, um, take it with grain of salt. I'm not sure that they are really unresolved, but I could check that. If somebody wants to know, please remind me and I will say. Type 4. This one is my favorite, simple because it's exporting DNA from T4, so it's very much related. Did you ever hear about Agrobacterium tumefaciens? That's the one that is used in genetic engineering of plants. So, and is exported through T4 secretion system. So, this one is quite unique and I love it. The secretion system is independent from second tag. So proteins or DNAs are traveling directly from cytoplasm to outside side of the cell without periplasmic involvement. So that's what distinguishes sec-dependent and independent. Um, Proteins that are related, so type 4 secretion system is exporting not proteins, I have to stop saying proteins, molecules that are related to bacterial conjugation, and they're not only exported to extracellular environment, they are also delivered to other cells. So, and this was very famous bacterium, agrobacterium, that is using that bacterium is using type 4 secretion system uh, to deliver again not proteins but DNA to plants. So, and this was used in genetic modifications of plants, but it wasn't developed like that. We we modified it, but generally this type 4 signal sequence secretion system is delivering DNA to the plant cell to cause specific plant disease called gut disease. So it's not only DNA, it the system is also secreting other proteins through type 4 secretion system. And there are, as you can imagine, specific structures for gram-positive and gram-negative bacteria. But what I want you to memorize from this one is that it is famous for Agrobacterium tumefaciens, and it's related to delivering of DNA material to cause gut to plants. Again, structures are different in a negative and positive bacteria. Um, so this one is characteristic by proteins that are called Vir proteins. And they can, again, as you can imagine, be B and D or many other different letters. But what is important to know is that they are Vir proteins. I won't be very specific. If you forget some letter, I really don't care. But just for your information, I'm giving you all these details in case you want to find some paper that is related to this structure. So, similar as everything else. It has inner membrane complex, which are BND Vir proteins, and there will be this specific flexible stalk. Uh, that flexible stalk goes from inner membrane, connects to the outer membrane, and outer membrane, again, we have proteins that are different than those in outer membrane. That's why I'm telling you just different types of Vir proteins from inner and outer membrane and flexible stalk, which again also has Vir proteins, just they are called with different, like 8 and 10, Vir BD and Vir BA. Again, Vir proteins will be enough and flexible stalk will be enough. Uh, there is extracellular pili, which is also Vir B. You can imagine why I don't care about memorizing all letters. It's just Vir proteins and flexible stalk and extracellular pili. Now, where the mechanism will come into the game is this um, fast-FIS transport, which is very similar. So this is one of those types that I really want you to understand. The first one was for type 1 secretion system MART toxin. Then we had the GSP DE transport. This one will be something that I really like to have. And of course, extra nothing for type 3. This one is interesting. This is figure from paper. Maybe figure will help, but I don't think it will be hard because it's called XT. So XT fat and XT fit. So it's a bacterial transport, bacterial toxin, which is a killer. So when there is no adhesives, like when there is no contact with the host cell, this fat is bound with the TP. And the way how I memorize CFAX is because there is this future energy system organization on campus that we all work with. So that's how I memorize. So FAS and FITS would be like a FITS. That's how I connect it. So when there is no adhesive, FAS and FIS are together. They're in complex. When adhesin is secreted, like when host cell is closed, so the contact is initiated, FIS is releasing fast, the toxin, and the fast XT fast toxin will bind to the ATPase. Again, notice that they are all Vir proteins. The energy that is released by ATP hydrolysis, so when toxin binds to the ATPase, that will cause ATP hydrolysis, and that will push the toxin to the peptidase. And peptidase will clear the N-terminal part. And one thing that I really found interesting, so when that peptidase leaves N-terminal part, that actually causes a remodeling of peptidoglycan. You guess this is negative. And when it remodels peptidoglycan, it makes like a passage for toxin to pass. Agrobacterium is DNA is much more simpler transport, but this was used in genetic engineering of plants. So what I was telling you about the transport of toxin, that the protein transport to C4 secretion system, this is DNA. So the initial step that precedes DNA transfer and DNA recruitment through the membrane. So you have to have something embedded in that membrane where DNA will pass, and this will be again something weird. So even though it's a different substrate for transport, it's still weird protein. So DFD is recruited to membrane, and once when that VRD is inside of the membrane, that's a signal for something called relaxosome, and I will tell you what that is in a second. So it's a signal for that complex of molecules called relaxosome to get activated. So what is relaxosome doing? So relaxosome is not one protein, so it's a set of multiple proteins, and what those proteins do, they bind to the origin of transfer, ORIT. And where is that ORIT? It is on the plasmid in agrobacterium, and that plasmid contains that infectious DNA molecule. So that's a basic, so this is literally conjugation. If you ever heard about bacterial conjugation. So relaxosome is here to initiate conjugation of transfer of DNA from bacteria to so, and this is all on the plasmid. tDNA is on the plasmid. So when TND, so this tDNA on the plasmid now contacts the inner membrane PRD, and now that relaxosome, which is bound to origin of transfer, can make the cleave of the piece of DNA from plasmid, and that piece strand gets, so that part of the plasmid gets like unbound because it's a DNA, so it's like oil. Uh, and it's pushed through the channel. So only part of DNA is pushed through the channel and it's cleaved by relaxosome. So this is way easier than protein transport. Type 5 is, in my opinion, the easiest one and the most easy to memorize because it's very unique in terms of outer membrane structure. So all of these secretion systems that we talk about have with all variations, very similar outer membrane structure. So you have outer membrane pore, inner membrane connector, inner membrane complex, and the fusion that connects to them. This one is very weird, but very unique. And it is also set dependent like type 2. That's not so complicated. There is no those GSP proteins involved. So these type 5 secretion system proteins are called, so transporters that are involved in with this system are called autotransporters, and you will see why in a few minutes. So what is this is so this is a potential mechanism because they're so diverse, it's not quite solved yet. I think it's because they're so diverse and each of them has a very unique structure. So the proteins that are going to be transported are interacting with amino acids in translocation domain, and that interaction helps protein to drive themselves through the pore. So that's why they are called autotransporter because they are driving their own transport. So it will be easier when we figure out what is the structure of molecules basically. So now we talk about molecules that will be secreted through the transport. So you have translocation unit, passenger domain, and then terminal signal sequence. So think about the protein that gets exported through this membrane as a three-part. N-terminus passenger translocation. So passenger domain is in the middle. And you can imagine passenger domain is the one that makes interactions. So now this N-terminus passenger and translocation unit have to interact with this general structure. So this is the general structure of type 5. So as you can see, there are so many different outer membrane structures. So even inner membrane gets variable, but general structure is this passenger domain, which is here, linker, which is here, and this potra domain. So potra domain is what really makes it so diverse. So again, molecules that are exported can be involved in pathogenesis or in biofilm. Mechanism, and again, that is showing how the diversity looks. So even inner membrane structure is quite diverse, but this potra is even more diverse. So they just have so specific and unique morphological structure. So what is the mechanisms? And again, proteins that will be exported to this system, they first have to be exported to SEC junction. So that's why this is SEC dependent, and that's why there is, so those that are first exported through SEC channel have that periplasmic event where they reside sometime in periplasm before they get exported. So once when they are transported, so now we talk about proteins, those proteins that have translocation units, passenger domain, and then terminal signal sequence. So now they're exported through SEC channel and they reside in periplasm. Protein is fully folded in periplasm now because the N-terminal signal sequence has to be cleaved. And again, we know it. There is N-terminal, passenger, and translocation. So that's now in periplasm. So this N-terminal has to be fully folded. With that translocator domain, protein for exports will interact with the passenger domain. So You remember that we have passenger domain of the receptor in the outer membrane. And what is interesting about this is once one interacts with this passenger domain, uh, protein for export is actually inserting itself into the outer membrane. That's why we call them autotransporters, because they energize themselves and they put themselves into the outer membrane. So this is very unique in the secretion of system. And translocator domain is actually energizing itself to be moved through the channel. So that's why we call them autotransporters. Type 6 secretion system is very, so this is all for type 5. Again, for structure, unique structure is what I would like to do. Type 6 secretion system is similar to type 3 and is discovered in Vibrio, but even though it's connected to Vibrio cholerae, which is pathogen, very often we see type 6 secretion systems in soil microorganisms like myxobacteria, because what those bacteria do, myxobacteria use type 6 secretion system to deliver certain toxins to the, so those bacteria are very socially involved, so they have something called outer membrane exchange system, and what they do through specific set of molecules, they identify their kins. So if they belong to the same group, they they have same receptors called receptors. I mean, that's not testable, but I'm just telling you to know. So when they interact with each other, if they have same primary receptors, they're not going to deliver toxin, but if they identify that there is something else that does not have same tri receptor, they will activate type 6 secretion system and deliver toxin through outer membrane exchange. And then that's how they will kill their non-kins. So they're very interesting social organisms, which I used to ask all the time, but I found it too much, so I'm not asking you to attend anymore. But they are interesting papers. I have some papers that I can share with you if you want to. So this is the general structure of type 6 secretion system. And again, similar as a needle. To me, this really looks like a phage. But generally, it's similar to the type 3 secretion system as a needle. So it does have similar, like a phage-like structure, but so type 3 is more like flagellum. This one is more like a phage. So this tail structure is something that resembles phage, and it has, you can imagine, ATPase and the tube, and base plate and the membrane complex. So, and through that structure, effector proteins will be delivered. Um, I am not really asking you to memorize these A and B proteins. I'm just showing you how type 6 secretion system looks. But I do want you to know that it is present in myxobacteria and is discovered in Vibrio cholerae. Just briefly, type 6 secretion system mechanism, this tail, like phage-like tail, that again looks like a needle to me. That's why it's very similar to type 3, will initiate the contact with the target cell, and upon detection of the target, like when they touch bacterial cell, the host cell, there will be ATP hydrolysis, and that ATP hydrolysis will drive complex formation of that sheet. Uh, and once it literally looks like a piston that exports protein to very similar to type 3 secretion system, like injectosome. So cocktail of enzymes, which are usually hydrolytic enzymes, and they will be different. There will be, it's not called injectosome, but it has very faint structure because those enzymes that are released through that, I mean, we'll call it page-like structure, but it's very similar to the needle in type 3 system. It is delivering those lysis proteins to the target cell, which is a host cell. Again, to me, it's very hard to make that distinction in between type 3 and type 6, even though they are quite different in terms of the structure, but the mechanism is very similar. Type 7 is Present, so this is the one that is present in gram-positive cells, and it's present in mycobacteria. You probably heard about Mycobacterium tuberculosis. So that's when studied in Mycobacterium tuberculosis, which is gram-positive. Um, this is all what I want you to know. Again, you will have ATP hydrolysis that energizes the transport. The structure is very different simply because we talk about gram-positive. So this is what I want you to understand, even though I'm not going too much in detail, because it's hydrophobic cell wall, like black and very thick. The whole mechanism needs to be different than in gram-negative bacteria. So this is studied in mycobacteria very specifically, and the mechanism that I am showing you here is very simplified, simply because I don't want to bother you too much. I already did. And this is the structure that exists in the inner membrane. It's called ECC proteins. And again, not really hard to imagine once you know how it looks. And you will see again there is ATP hydrolysis, and there is chaperone called ECCA. So again, membrane components are ECC, G, and C. So and there is a B that's kind of extensive to the small, teeny, tiny periplasmic space. So when the protein that you are exporting, which is usually some pathogenic like protein, with about to be transported, ECC, which is here, is completing hydrolysis. So ECC is ATP hydro analog to ABC transporter. So it has hydrolytic activity, and it is associated with the inner membrane. And that energy from ATP releasement is what is driving a protein through the pore. Um, the outer membrane component, that I shouldn't even call this outer membrane really, because it's more like that hydrophobic part in gram-positive cells. That it says it's not resolved yet, but again, I am not in the field of pathogenic microbiology, so if somebody knows that the structure is resolved, please let us know, because I am not really aware if it got resolved. Maybe that is something that we can search. Like what's the structure of outer membrane or cell wall component? And substrates are different antigens, different molecules that will be associated with pathogenesis. And this type 9 secretion system is, I always suggest people to uh, pick type 9 secretion system as their paper for this class. Nobody ever took it, which is too bad. Because when I made these presentations, it was characterized in 2008, which was relatively recently. So there was only one paper that I found, and it was a review paper so it was review on two papers, which I didn't think it's enough to make a review. It's very young, it's very new. All what I know about this is that it has a large signal polypeptide, which is like transmembrane domain, and the four with a cap on one end and another opening on the other end. Um, Proteins that will be delivered are entering the pore, and then cap is closed, and then channel 4 opens on the other end, and proteins get dissolved, get exploded. I don't know much about it. So if somebody wants to take a paper to present of type 9 secretion system, that would be highly beneficial. Um, just nobody ever took it. So again, very interesting topic. In terms of Archaea, there, as usual, I don't have much to say about Archaea, which is not because they are bad, just because I don't know much about them and I don't work with them, and nobody works with them, so nobody really cares. Which again, they're good. So we talked about archaeal transport systems when we described archaeal biosynthesis in Unit 1. So that's the only secretion system that I am aware about in archaea. So those base point ATPases, if you remember, that they look like a nice little triangle that get exported through the specific structure in the membrane. Uh, yeah, I don't know much about that here, which is just because I am not working with them. And I will stop here because then I have literally only one slide. There is nothing too much. But what you need to know is this. who, what, how. Who is transporting type of the system, what is transporting which proteins and how mechanisms. And again, when the exam comes, I will tell you exactly which signals you need to know. And that's all.
Take pizza if you would like it.
If nobody takes it, bring it to the